What is translation? Yes, the term may describe explaining something in a different language, but I meant more as a term in biology. Translation is the formation of protein or polypeptide from an mRNA strand. An mRNA strand is a strand made from the information derived from the DNA, or more specifically known as deoxyribose nucleic acid. Think DNA as like a big recipe book, like really big. So big that you can come up with almost 150 million combinations. It would take me forever to choose something. Luckily, our body knows what it wants, and it also knows how it can get exactly what it wants. For example, if you eat something, our body processes it into sugars using proteins. More specifically, a group of proteins called enzymes to break this down into sugars. So this is just one of the many functions that a protein can do. But in order to get these magical proteins, one must go through the tedious but wonderful process of transcription and translation. Today we're going to go over translations in eukaryotes. Eukaryotes are multicellular organisms, and prokaryotes are single-cellular organisms. The translation process in prokaryotes is similar to the translation process in eukaryotes, except the mRNA strand that the prokaryote uses for translation is what is called a polycistronic mRNA, which means it is an mRNA strand that has more than one binding site. But to talk about translation in eukaryotes, we first need to go over something called tRNA. tRNA, or transfer RNA, transfers something called amino acid to the ribosome. Amino acid is a compound that carries an amino group and an acid group, just like the name indicates. Amino acid are what proteins are essentially made of. They're like the building blocks of protein. Ribosomes need to glue these amino acids together to make a protein, and tRNA delivers these amino acids. There's all sorts of tRNA, and two tRNA are not the same to each other. Like two tRNA, they will never equal each other, because the ribosomes can recognize which tRNA has the amino acid that they need. It's like if you order a pizza, you don't want a vegetable pizza from Pizza Hut, right? You want a Hawaiian pizza from Domino's. If all the pizza delivery people wore the same uniform with the same logo and the same brand with the same pizza box, you wouldn't know if you got the right pizza from the right place. Also, if you don't think pineapples don't belong on pizza, you are a heathen. Anyway, the tRNA has 3 loops, and it sort of looks like a clover. Here's a picture of one. It also has 4 double helical segments when it's folded. By folded, I mean that the tRNA literally folds upon itself. This happens as the amino acids basically huddle together to create these really strong hydrogen bonds. They do this to determine the protein's function. And the protein folds into a certain shape, so it needs minimal energy to maintain that folded state. The tRNA has 2 forms, when it is unfolded and folded. The tRNA is read from the 5' end to the 3' end, and the distinct loop at the 5' end is called the DHU end, and the distinct loop at the 3' end is called the T psi C. The psi in T psi C is the weird trident looking symbol. But the most important area of the tRNA is at its bottom, which is called the anticodon. The anticodon is what determines which amino acid the tRNA is going to carry, because it is complementary or matching to the mRNA sequence. This is written or coded from 3' end to the 5' direction, which means it was transcribed from the 5' end of the template strand. The 5' end has the phosphate group, and the 3' end has the hydroxyl group. So, the tRNA needs to deliver this amino acid, but to do that, it needs the amino acid. Remember, nothing in life comes free. So tRNA needs help in getting this amino acid, and a family of enzymes called amino acyl tRNA synthetase does exactly this. It attaches itself to the DHU loop of the tRNA, and using the energy from the ATP hydrolysis, which is the reaction that occurs to remove a phosphate group from ATP using water, to attach the amino acid to the amino binding site, or more specifically, adenine from the amino binding site sequence on the tRNA, which is located near the 3' end of the tRNA. So let me repeat that. So, the family of enzymes called amino acyl tRNA synthetase is what attaches the amino acid to the tRNA using ATP hydrolysis. So, after that happens, the amino acyl tRNA synthetase detaches itself from tRNA, and the tRNA is now in its charged form, which is the term to describe tRNA with amino acids. Now that the tRNA has the good package, it can truly start translating. In the first step, the transcribed and modified mRNA comes into the cytosol of the cell, which is essentially the space outside of the nucleus. Their translation begins. So the initiation factor binds to the 5' cap, or the end of the mRNA strand. An initiation factor is basically a protein that has one and only goal, which is to initiate translation. After the initiation factor binds to the 5' cap of the mRNA, the small subunit, not the whole ribosome, mind you, only the small subunit. The small subunit labeled 40S is recruited or added to the initiation factor. At the same time, the charged tRNAs, remember those? So the charged tRNAs, which is the tRNAs with the amino acids, they see the ribosomal unit getting initiation factors and decide that they want some initiation factors too. So the charged tRNA also binds to an initiation factor. But this initiation factor of the charged tRNA is special because it has something called a GTP, bound initiation factor. So GTP is an energy carrying molecule which is kind of like ATP, but it doesn't have adenine, but instead it has guanosine or guanosine. In the third step, the tRNA that is charged with methionine binds to the P site of the small subunit, which is the middle area of the subunit. A little side note here. The ribosome has 3 main sites for binding to occur. The first site is the exit site. The middle site is the peptidyl site. And the last site is the amino acyl site, where the incoming tRNA can bind. Anyway, where was I? The tRNA with methionine binding to the P site of the small subunit. So after the tRNA binds with the subunit, the whole complex, now composed of small subunit initiation factors and a charged tRNA molecule, wants to find the start codon AUG in the mRNA strand, because the charged tRNA with methionine has the anticodon that matches with the start codon AUG. So the whole complex starts moving until it finds the start codon AUG. Once it finds the start codon, the complex becomes excited and changes its shape. This change causes all of the initiation factors that are attached except one to lose its affinity to the complex as the GTP that they are bound to gets hydrolyzed into GTP and get released. Basically, they lose all their energy and don't hang around anymore. Only one initiation factor in the A site of the initiation factor gets left behind, but we won't worry about that for now. Finally, the large ribosomal unit can join in the fun, but it needs a wingman to bind to the complex because itself alone isn't enough to join the cool group. So it gets help from a GTP-bound initiation factor. The factor binds itself to the large ribosomal unit and binds the large ribosomal unit to the complex. After the large ribosomal unit successfully binds itself to the complex, it gets released by hydrolysis, but not before taking the remaining initiation factor in the A site with it. So after all of that, now we have a complete initiation complex with its sites all open for business. The initiation complex now has the ability to start a process called the elongation process. So I'm going to summarize all of that. So to summarize all of that, number 1, the initiation factor binds to the 5' cap of the mRNA. Number 2, the small ribosomal subunit basically binds to the 5' cap end and the initiation factor. Number 3, the charged tRNA gets jealous of the small ribosomal unit and gets the initiation factor for itself as well. But this initiation factor is special because it has a GTP bound to it. Before the large, number 5, before the large ribosomal unit can bind to the complex, the initial the whole complex, which is the charged tRNA, the small subunit, and the initiation factors, they move alongside the mRNA sequence to find the start codon. Number 6, once it finds the start codon, what happens is that the complex undergoes a conformational change. It changes its shape, so the initiation factor on the tRNA loses affinity, and every other initiation factor also loses affinities because of the hydrolysis of GTP, and it all gets released except one initiation factor in the A site. After the ribosomal unit binds, we are left with the complete initiation complex, which is now made up of 1, charged tRNA that has methionine, number 2, a small subunit, number 3, a large subunit, and number 4, the E, the P, and the A site. The E and the P and the A site, which also stands for E, the exit site, is where the tRNA is going to leave, and the P site, the peptidyl site, is where the middle area, the P site, is where the tRNA is going to reside in, and the A site, which is the arrival site, well, officially it is called the amino acid site, but it's more helpful to remember it as the arrival site because that is where the tRNA will arrive.